All right, so we are starting Unit 5. This is going to be DNA, RNA, protein synthesis, genetic technology, and that's in a little bit of genetic mutations. So the idea for these notes for today is compare and contrast DNA and RNA. We want to make sure you understand what base pairing is within N bases, as well as we needed to do a DNA code and sequence for protein synthesis. Of course, what is DNA? So every new cell that develops in your body needs an exact copy of DNA from the parent cell. And if this is again body cells, so if it's an exact copy, obviously we remember that was mitosis, right? But we do need DNA in every cell, exact copy. All organisms must be able to pass their copies of DNA to their offspring and basically to continue the species. And remember that would be called obviously meiosis, right? So your body cells need the exact DNA copied through it, and all organisms must be able to copy that DNA for new species. So with the structure of DNA, we do have two primary functions of DNA though. First one is to store and use information and direct all the activities of the cell. The second thing is it needs to copy itself exactly for new cells that are created. Okay, so it stores and uses information and directs the activities. Your DNA is basically who you are, so everything is ran by DNA. Okay, so then also again it copies itself for new cells that are created. All right, so earlier in the semester we learned about DNA structure. So yes, we still have the phosphate, and because this is DNA, this is deoxyribose, and then this right here is the N bases. So DNA again are made up of nucleotides. These three things are the nucleotides. So just to review the nucleotides again, it has DNA has a 5-carbon sugar, and that sugar is deoxyribose. DNA. This is where the D comes in because it's deoxyribose nucleic acid. Okay, so deoxyribose. For anything that ends in ose is a sugar, and for DNA, this starts with a D. So this is how you know that's a sugar. Then it has the phosphate group, and remember it has the nitrogen bases: adenine, guanine, thymine, cytosine. Now, DNA is two long strands, and remember it's shaped as a double helix, or sometimes we call it a twisted ladder, and that's the twisted ladder of nucleotides. So the sides, which is this, or this, as you can see, which is the pentose, is the sugar molecule. So that sugar molecules is deoxyribose. Now the N bases are here, or what we would call here. The N bases would be adenine, guanine, thymine, cytosine. So with the nitrogen base pairing, remember adenine always goes with thymine. Remember I told you before, apple tree, right? A goes to T. And this is held together by two hydrogen bonds. Guanine always goes with cytosine, and then I always said this is garbage can because G goes with C, and this is held together by 3. The reason why is hydrogen bonds are very weak. So here, this is just a picture to show you the twisted ladder, and again, the sides are sugars, the inside is the N bases. And again, A goes to T, G goes to C. As you notice, the sugars are now connected by what? The phosphate. Now, who actually discovered DNA? Now, the thing is, DNA was not created in 1953. It was discovered. We always had DNA. So in 1953, we actually had two scientists to figure out the shape of the DNA molecule was a double helix. So, and this happened with Watson and Crick. Rosalind Franklin, who was a woman, was actually now recognized as part of the process, as you see here. So now that we understand the DNA process, and we understand that DNA can only match what an apple tree and garbage can, we're going to go now into protein synthesis. This is your key point, all right? Because the process is that we have an original strand of DNA that will split and make two strands of DNA. All right. So the first one is replication. So replication, what do? We learned that, remember, in mitosis and meiosis that replication take place in interphase, and it was taking place in the S phase, right? So DNA makes a copy of itself in interphase in the S phase. So remember I told you, like Simon says, so you do whatever Simon tells you to do. This is replication of DNA. This process is, again, DNA replication. It's with the help of enzymes that unzips the hydrogen bonds. So here, the hydrogen bonds that connect those together, what happens is now it's unzipped, just like a zipper. Because if you put a little zipper here, like act like this is a zipper, this will close. So we unzip it by the hydrogen bonds. Then the complementary nucleotides will come together and eventually add, as you see here. All right? So what are the complementary base pairing? Well, we're going to have old, or the original portion, is the template to make the new chromosomes. Okay, this serves as the template to make the new chromosome. So using the complementary base pairs, the end will be two DNA molecules identical to each other. So again, it's one to make two. All right, so A goes to T, apple, tree, garbage, can. All right, so what I'm going to do is show you what that will look like. So what would a complementary base pair look like? So on your EOC, they will have a strand of DNA, and you would have to, you make sure you know what it's saying. First, this says complementary pair. Anything that's a complementary, it complements each other when we're talking about DNA. So this is DNA. So apple tree, garbage can. So that means C is going to go to G. G goes to C. A goes to T. T goes to A. T goes to A. A goes to T. C goes to G. G goes to C. T goes to A. All right, so I need you to write this in your notebook. I need to see this in your notes. This is important. You need to understand complementary. All right, so now we did all that talking about DNA. What about RNA? Okay, so of course this is DNA. It has obviously double helix, two-stranded. Um, this is a one strand. It's missing a whole side. All right, and you notice here we have the T's, but this one only has U's. So what is RNA? RNA is ribonucleic acid, and it has the function of synthesizing proteins. Okay, this is what synthesizing means, to make. So what is the purpose of RNA? It is to make proteins. Okay, ribonucleic acid. Well, what makes proteins as a cell organelles? Ribosomes. So this is where RNA is coming into play. Okay, and this is made from the replicated piece of DNA, not the first strand or the original strand of DNA. It is made from the replicated piece of DNA. So how does this take place, Miss Cheek? Well, again, RNA only has one strand of nucleotides, has ribose as its sugar instead of deoxyribose. So RNA to me is just like, okay, so DNA is like Jordans, right? And RNA is like Shaq's. Got it? Okay. So instead of having thymine, it has uracil. So there's no T's in RNA. No T's in RNA. So what are our base pairs though? That means A goes with U and C is still goes with G. Okay. So A, U, G, and C is still together. All right, so A is goes with U. There's no T in RNA, and C goes with G. Now, RNA is different in the DNA in 3 ways. So you have to identify the difference between DNA and RNA. Well, one of the first differences is one-stranded. Okay. Next one is the sugar. RNA has ribose, DNA has deoxyribose. And the third thing is that RNA does not have thymine, it has uracil. Please remember, I'm putting a star on here because this is very important. All right, so now there's 3 types of RNA. 3 types of RNA. The first RNA we want to talk about is messenger RNA. So with messenger RNA, this is the RNA that serves as a template for the assembly of amino acids. This is how we make our proteins, because remember, amino acids make up our nucleotides for proteins, right? So mRNA is going to get the amino acids in the right order to make those proteins. This is what you have to know, that it leaves the nucleus and goes to the ribosomes, because RNA is ribonucleic acid, right? So that ribo. Then it goes to the cytoplasm, where the amino acids are assembled into proteins according to the instructions. And I know it fully may not make a lot of sense, but think about it. Messenger. When we do to text message, right? Because this is part that's going to call transcription in a minute. When we write text messages, a lot of times we be like OTW, BRB, LOL. Those are 3 letters, right? We're sending a message and it's in a code. And this is what's going to start making sense. We have transfer RNA. We also, this is nickname is tRNA. It is folded back strand. Okay. So what happens with this? It kind of grabs the amino acid. Exists in 20 different varieties. Why? We have 20 amino acids. So it has to be in 20 different ways. This delivers the appropriate amino acid to the ribosome. So what takes place is that this little puzzle piece will grab on, because sometimes it looks like this, like on, let's erase that. But it'll look just like how it is here. It'd be like a little puzzle piece, and this puzzle piece will grab on to the amino acid, which is in 3 little codes, and it will move it to where it needs to be. Okay. So then we have ribosomal RNA, which is called rRNA, which is a globe-shaped structure. And this right here on the ribosome, it helps peptide bonds form. Now, so remember, what's the whole point of protein synthesis? To make proteins, right? And it has to do a lot with the ribosomes, because ribosomes make proteins. But what type of bonds was there? Remember, the amino acids made peptide bonds. So this is how the peptide bonds are filled in amino acids. And then, but the thing is, the amino acids have to be in the right order. So what's the next step? The first step was replication. Step number 2 is transcription. So this occurs in the nucleus. Transcription means to write. So remember, I was talking about messenger RNA, and we had to write a text message, right? So mRNA is made from DRNA. Lord have mercy. mRNA is made from DNA. The new strand of DNA is unzipped, right? So remember, we have the DNA and it becomes unzipped. And there's an enzyme called the RNA polymers. What happens is that RNA polymers uses the complementary base pairs. The N will be the old DNA molecules in a new piece of mRNA. And as you see here, this is the DNA, right? It split it, unzipped, and went into two pieces. So what happens is the RNA slips in and it begins to connect. Remember, A goes with U and C goes with G. There's no more T. So transcription means to write. And how do we know what's transcription? Message, because you're going to write a message. All right, so the new piece of mRNA now leaves the nucleus, goes through the nucleopores, goes through the cytoplasm. Why? Because our final destination needs to be to a ribosome. So remember, this is the site of protein synthesis. And what is protein synthesis? To make proteins. We group this, though, by every 3 N bases. So why did I say like OTW? Why did I say LOL? Why did I say BRB? Okay, if I say right here, this right here are 3 letters, right? And we sometimes have a tendency to type text in 3 letters because it's a code. So this has 3 bases. So you see here, it's grouped in AUU, CGC, ACG. These groups are called codons. This is code. If I text my grandma HRU, she gonna call me and be like, Shantae, like, what is this? So that's the code. What happens is in transcription, we send a message, and a lot of times we send text messages, we send text messages as codes. And so this is called a codon. It's a group of 3. Each, again, is a code for a specific amino acid. How do I know what type of amino acid it is? It's going to be in the next step. So we're actually going to use a codon wheel to determine the amino acid that will be delivered to the tRNA, okay, which is next. The tRNA will make sure it gets where it needs to go. It's like a taxi. That's basically what the tRNA is, because it's called transfer RNA. So the codon wheel we only use with mRNA. Why? Because that is the message, and that's the code. So this is the mRNA code. So when you have to get the first thing that we do is we have DNA, right? On your test, it's going to have a DNA strand, and it's going to be like this. It's going to be written in 3s. Once you get this right here, it's going to have something like this, mRNA. What do we do? Well, T goes to A, and A now is going to go to U because it can't go to T because mRNA doesn't have that. C goes to G. So now once I get my, I did not know that I misput it in. Once I get my mRNA, okay, which is AUG, that's my first code, I have to find it on the codon wheel. But how do I do that? Well, start from the big letter, A. Next letter was U. U. Next letter was G. See how it kind of just went down? Well, that name is methionine. I'm just going to write meth. So that is my amino acid. Okay? So DNA, we're going to use that to get our mRNA. But the amino acid comes from the mRNA because of here. So this is basically like a book to understand those text messages. Like, this is the book that your grandma will pull out and be like, she just sent me OTW. What does that mean? And so we open up the book and we go OTW. Boom, we find out what it means. Well, maybe you can't use that chart. Maybe that was too hard. Maybe this is a little bit easier. So I'm going to use just a random one. GAU. Now, first of all, you don't see a T, right? If you ever try to look up a T, you know you did something wrong. So GAU. So again, Miss Cheek, how do you do this? Well, first letter, so G. Second letter is A. So I know I'm going to be in this group, but obviously I'm in a G, so I'm down here. And then my next letter is U, and I see U here. So by the time I meet it, G, A, U. Aspirate. All right, that's how you do it. So now let's get to our third step. Third step, okay? So we had replication, we had transcription, and we had translation, right? So basically, as I told you, this occurs in the cytoplasm, but this is when the mRNA attaches to the ribosome, and this is after we use the codon, it goes with the anticodon, which is tRNA. So again, these is the if it ain't Shaq, these we really the Walmart brand. Okay, tRNA picks it up. See, as I told you, I drew that picture earlier. It picks it up and takes it to where it needs to be. So once mRNA goes and codes, okay, just like we did on the codon wheel, tRNA says, okay, let me go ahead and pick it up. tRNA translates this to the right thing. So once it transfers, as you see here, because it's part of translation, tRNA transfers. I'm going to say tRNA is my taxi. It picks up my amino acid and brings it to the right place, the ribosome. And that's when the bonds will form. And this is where the translation takes place, because they got to be like, what did she say? Okay, now I'm taking you to your destination. Okay, you on the way? Yes, ma'am. All right, so that's kind of how that works. So to sum it up, a protein may consist of hundreds and thousands of amino acids. It's only 20, so it's a lot of order. Which codes again for a specific protein. These amino acids can be arranged in particular sequences, and that's the only way they work. And then we have structural proteins that are in plasma membranes, and then we have functional, which is in your hormones and enzymes. So how does the protein synthesis work again? Remember, DNA replicates itself, right? So this is DNA to DNA. That is when apple tree garbage can. What is transcription? DNA to RNA. And which RNA? mRNA. Okay. Then we have, and we use the codon wheel to get the amino acids. Once we get that, we have translation. Remember, now this is RNA to RNA. Now, again, more specifically, we have M to your T, and the T is going to take it to your rRNA, because it's the last thing, right? Ribosomes make protein, so we have to get it to the right protein by getting it to the right ribosome. So M to T to the R. All right, remember that's tRNA is like our taxi. So let's try to do a problem together. This is called simple protein synthesis. So remember we get DNA, and as you see here, this is our coding strand. So the strand that you usually get on your EOC, it will be specific. It will say, hey, go, this is a strand of DNA, make a complementary strand of DNA, which will be this one, okay? It may say, here's DNA, which is already, honestly, when they give you a strand of DNA and tell you to go to mRNA, it's already been through the replication area. So DNA to mRNA, and then we have to get the amino acids, and then we have to make sure you understand how to go from mRNA to tRNA. So I'm going to show you that. Please write in your notes. All right. So ATG. Well, let's make replicated DNA. A goes to T. Let me just choose a different color for you. A goes to T, T goes to A, G goes to C, right? That's our first one. A goes to T, T goes to A, C goes to G. Second, T goes to A, C goes to G, G goes to C. Last one, T goes to A goes to T, A goes to T. All right. So now I just made replicated DNA. Now this is going to be a little bit off. So now I'm going to do my mRNA in a different color. T goes to A goes to U, C goes to G. Code it. You block it off. T goes to A goes to U, G goes to C. Code it. A goes to U, G goes to C, C goes to G. And my last one, hopefully I can, U and T, A. All right. So now I need to get my amino acids. Let me use a different color again. So now what I want you to realize is that you see how it is pretty much going in order. So now with the amino acids, we understand that I have the code from mRNA. And now this is our codon wheel. Hint is every DNA strand, in order to really start, must start with AUG all the time. AUG means start, which is methionine. All right. As you see here, I know this is small, but AUG, that's methionine. So I'm writing down my amino acid. And again, I'm going to make a little arrow so you can just kind of make sure. And next one is AUC. I can't see the full word for it, but it's here. I can't tell you which one it is because I don't have my thing. And that is my fault. But the next one we do have is UGG. UGG. And it says tripe. So I'm just going to just write the first part. And then it says UAA. And what's this word? It says stop. So my DNA meth always means start. So your DNA will tell you how long to go. Your DNA is not supposed to stop going until it says actually stop. And there's 3 stops. Now, once we get here, we got our amino acids and we're good to go. But on your EOC, sometimes they're going to ask you for tRNA. Well, how do I get tRNA? You see here what you have from mRNA. We got to go from there. So A goes to U. U goes to A. G goes to C. And I'm getting this from the mRNA strand. So A goes to U. U goes to A. C goes to G. I'm just blocking these off so you can. And then U goes to A. G goes to C. And then here, A, U, U. And that's how you get them all. So if you ever had to go backwards and it says go to tRNA from to mRNA, you would say, well, U goes to A goes to U because you're still doing RNA. If it says go to mRNA to DNA, you will go A goes to T, U goes to A. This is just a question. Here's a codon sequence. This is like a question on the EOC. It will give you something like this, and it will say, hey, what is the amino acid for this code? What you'll do is go on the codon wheel. Here's another question. If they gave you this, and a mutation of it would be, we haven't talked about mutations yet, so don't worry about this question. But this will be AUG. I told you it was always meth, and you just keep on going from there. Okay, I don't. So you would have a codon wheel, and you would have did this on the test. An amino acid has a sequence of alanine, histidine, and arginine. Would have what codon sequence? So you would have to go to the codon wheel and find these words, and then you will write down the 3 letters for this words. Sometimes it would say go to the codon wheel with these and give me the DNA strand. So of course you would just have to work backwards. GCU, and given the codon wheel and you given the sequence of UGG, what amino acid would that be? You would just go to the codon wheel and figure it out. I know these notes was a bit long, is because again this is a very, very important part um, of our lesson and on the EOC. Hopefully you can understand this. If not, I'm gonna be doing more practice with you.